So I'm going to be fairly brief. I'm really going to give you an update. I'd like to give you an update on two things. As Long has mentioned, he is our medical director now at our clinical research sequencing platform, so I want to give you a little bit more information on that. If you came to the Illumina workshop yesterday, you would have heard Sheila Fisher give a great overview of the genomics platform as a whole, of our technology, our install base, and the numbers, the samples we work through. So I'm not going to belabor that, but suffice to say, obviously, we have a large amount of research sequencing in our research platform. We run genomes, exomes, RNA, many, many different products. So what I'm going to really give you a little update on is just our clinical lab. So about a year and a half ago or so, a group of us at the Broad were tasked with doing a feasibility study and understanding if there was interest and a logical reason for us to start doing clinical sequencing. And it actually was a fairly easy assignment because more and more of our collaborators, our PIs, and folks who come to us externally are doing clinical, preclinical, translational kind of work. And so we thought there was actually a great value to take what we've built in the genomics platform on research level sequencing to codify, document, validate, and put some of that into a regulated environment so that folks coming to us could also get the advantages of what we offer in terms of process, efficiency, and quality, but have it in a CLIA environment. And so we thought this was a great way for us to make available all of those tools, both process and analytical, to the community in a CLIA environment. As Long as mentioned, we're CLIA licensed in the state of Massachusetts. We have recently had our first CAP inspection, and we expect accreditation in the coming weeks. Right now, we have one validated assay, which is basically a whole exome sequencing assay that is coupled with our traditional high-quality analytical pipelines, which includes Picard alignments and GATK germline calls. We are currently working on Gadiget's Firehose suite of tools, so we can do tumor normal somatic analysis as well. One differentiating point for us, I guess, to other groups who have built clinical labs is that we really thought that it made sense for us to focus on our core competency, and we believe that is in high-quality data production at whatever scale is required, and to apply new techniques and validate those for clinical applications. But that our community, our Broad community, and as you can see from this meeting, a lot of groups around have put a lot of effort into dealing with NGS data from an interpretive standpoint. The genomics platform is not in a hospital. We don't have clinical fellows. We have Long, but we don't want to burden Long with interpretation of thousands of exomes. So we don't offer interpretation. We really focus on our core competencies of generating high-quality data, and then there are many groups in the local hospitals and around who can take that data and do the clinical reporting. Part of this mission of CRISP, though, is also to take on challenges, just as Long as described. There are many diagnostic and clinical research challenges that can be addressed by the application of new technologies. And so looking at clinical assays, NGS assays specifically, is something that we're very interested in CRISP. So the first challenge that we got came from some collaborators at Dana-Farber Cancer Institute. Ben Ebert came over to us and gave us the story of AML and leukemia in general. I am not an oncologist or a clinician, so a lot of this information comes from then, but clearly AML, for those who know about it, is a terrible diagnosis. It has a rapid time course and a very short survival if untreated. So there's a clear clinical need there. And one of the challenges with AML has been the requirement for a rapid and efficient identification of patients who may benefit from a novel genotype-directed therapeutic. So just as Long has outlined in the lung cancer cases, there are clear benefits to having a rapid diagnostic for AML, so you can quickly enroll people on novel therapeutics or trials, and they don't have time to wait, basically. The challenge to us was we need a really rapid way of figuring out SNVs, indels, and clinically meaningful fusions in these patients, and we can't really wait for hybrid selection. You can't wait 3 weeks to get the exome back. There are a small enough number of targets here that we want something targeted, but we want it fast. And so that was where we started from, and it just so happened we had been talking, I had a long-standing relationship with Jason Myers, and he told me about this assay that Long had developed, and our interest was piqued. And now, as Long has mentioned, there is actually versions of that assay available for SNVs and indels, but what I'm going to focus on is just the application about fusions, because that's what we had looked at. Just as in the lung cancer case, AML has a large set of characteristic fusion events, particularly in this one gene, MLL. I've seen many posters downstairs that look at various aspects of MLL. This gene actually has up to about 79 currently, I think, characterized translocation partner genes. So this is a case where clearly doing targeted design of traditional primer design would be limiting, would be difficult, and there's a lot of potential partner genes there, so that would be a limitation. And the other really cool thing is that some of these fusion events are actually actionable. Maybe it's only in the prognostic case, but in the more and more there are some therapeutic events, the BCR-ABL clearly, and MLL also has some potential therapeutics. So really being able to characterize rapidly an MLL fusion would be beneficial for these patients. So that was what we came to Jason and Enzymatics with. And I won't go through the technology long. We did a really good comprehensive view of that, but clearly in the description there are many advantages to this, not having to know the exact breakpoint, being able to tile on a single primer, and the rapid processing time. So our initial pilot was basically, we had the Archer pipeline design primers, both the 3' and 5' direction along the MLL gene. We, with our collaborators, Lindsay Coleman over at Dana-Farber, identified 9 cell lines that had characteristic AML and ALL fusions. We ran the assay at Broad with kits that Archer provided us. These were actually pre the lyophilized kit, so our kits were a little bit more manual. We ran all the samples on a single MySeq. We ended up with about 1 million reads per sample. We then gave the data back to Archer to run in the Archer analysis pipeline and took the data back from them. And then we also independently took the raw data just to do our own sort of independent confirmation, to do realignment and make sure that we could also see what the Archer pipeline was producing as a report. And so we're happy to report that actually in all 9 of these cell lines, which all had characterized fusions, the AMP assay came back with the expected primary fusions. The AMP report, as you'll see if you use it, has fusion candidates 1 and 2. In one case, the primary fusion candidate was candidate 2, but there were very similar number of supporting reads. And you can see here actually the interesting fact about, we had about 1 million reads per sample, but the fact that we were able to deduplicate using this random priming means you can actually identify the unique reads, and you can see by the lower number of reads here how much PCR duplication there actually is. From our own independent visualization, we just took one of the cases here, so you can see the MLL and AF4 fusion partners color-coded on here, and you can see again the characteristic random starts. So that was really encouraging. So it was a simple pilot. That's where we were right now, but we're pretty excited about the assay, the technology, for all the reasons that Long mentioned. To do the MLL assay in particular, we'd want to now take this onto real clinical samples to do a traditional validation for sensitivity, accuracy, and precision, et cetera. I am particularly excited about the information coming out of Enzymatics about their DNA panels, also about larger fusion panels that are more pan-cancer type panels around heme cancers and solid tumors, and also the fact that it works from FFP. We see a large interest in integrated analyses of DNA and RNA from FFPE samples going forward, particularly in the clinical space. The lyophilized reagents and the workflow improvements, of course, are really, really exciting about in terms of workflow and rapid turnaround of testing. So that's where I'm ending. So basically, I just want to thank Jason, Long, everyone at Enzymatics, everyone at the Broad who ran this, and our clinical collaborators at Dana-Farber. If you have any interest in or any questions about the clinical lab or the genomics platform in general, genomics@Broad Institute is our sort of group email address, so please feel free to reach out to us. Thank you.